phosphorus or losing them?
That's one way. I'll tell you the way I look at it. If the ATP is at the head of the arrow, it's energy pale. If the ATP is at the butt of the arrow, it's energy investment. Okay? So, if you see this, okay, that's investment. If you see this, that's pale. Okay?
Can you just say when pyruvate is pale?
Well, if I give you pyruvate name, then yes. But also notice up here, we're making phosphoglycerate, 3-phosphoglycerate. And that's technically part of the payoff phase as well. This whole thing here is the payoff phase. So instead, I just look at the arrowheads. Okay, I see the arrowhead and it's pointing at the ATP. That's pointing to pale. Back here, the ATP is by the back of the arrow, the posterior of the arrow. That tells me it's energy investment. It's a simple way, it doesn't require a lot, and I think it's just something easy to remember. Now, we've made our pyruvate. Remember, we've made 2 pyruvates, and here's the neat thing. Those pyruvates are going to actually be used in the citric acid cycle. But the problem is we can't actually use pyruvate in the citric acid cycle. We have to first convert it. So as if it wasn't crazy enough what I'm talking about today, you know, as if it wasn't bad enough that we're now transitioning from glycolysis to the citric acid cycle, during that transition, pyruvate is also going to be altered. So we're actually going to have a reaction occur before we get to the citric acid cycle that converts pyruvate into a molecule that's called acetyl-CoA. Basically what happens during this process is we're going to remove carbon dioxide from pyruvate. In the process of removing that carbon dioxide, we're also going to remove some electrons. So we're going to produce more NADH. And at the very end, we're going to bind the remaining 2-carbon compound to a specialized enzyme called, or coenzyme called coenzyme A. Sure. Let me give you a picture because I think it will work better with a picture. Okay. So we're going to take pyruvate, a 3-carbon compound, and as we move it from the cytosol into the mitochondria, it's going to move through this transport protein, and as it does, we're going to remove a carbon dioxide, so this CO2 here is going to go away. It's going to be removed from the pyruvate. In the process of removing that carbon dioxide, we're also going to then begin removing electrons from this pyruvate molecule. So we're going to remove 2 electrons and hydrogen, which is going to produce NADH plus a free proton. And we're then going to bind coenzyme A to the 2-carbon containing compound. This right here, this transport protein, off the top of my head, I want to say that it's actually passive transport because we're not using any ATP. Yes?
Did you say bind to coenzyme A?
Yes. Okay. So these 2 are weakly interacting. Okay. That's what is represented by that kind of squiggled bond, is that acetyl-CoA is interacting with coenzyme A, but they're also not showing you the full structure of coenzyme A. And really, all this is, what you're going to see is coenzyme A is just acting to get acetyl-CoA into the citric acid cycle. So once this 2-carbon molecule is into the citric acid cycle, coenzyme A is released. Okay? So again, we remove carbon dioxide, we reduce NAD plus to NADH, and again produce another proton, and then we attach coenzyme A to the 2-carbon molecule. Questions?
So CoA's only purpose is to be converted to CO2?
No, COA's purpose is to attach to this 2-carbon molecule and get it into the cycle. At this point, we actually haven't even entered the citric acid cycle, but if we did not produce this acetyl-CoA, we can never get into the citric acid cycle. Okay? So, we need to remove this carbon.
Getting into the cycle.
What's that?
So, yeah, never mind.
Yeah, this whole process is to get us into the cycle.
Okay.
Yeah. Yes, Allison?
Sorry, you said remove what? Said, like, in the beginning, like, what do you remove?
In the beginning, we're going to remove carbon dioxide. Okay, so at 1 here you see we're removing carbon dioxide. We're then reducing NAD plus to NADH plus a free proton, which means we're oxidizing the remaining 2-carbon molecule so that it can bind to acetyl-CoA, to coenzyme A to form acetyl-CoA. We need to form acetyl-CoA or we can't enter the citric acid cycle. Okay, so if we don't produce this, we're not getting into the citric acid cycle.
Where does it go after it gets into the cycle?
Once it's in the cycle,
Then it's converted?
coenzyme A is removed, and we're going to see this 2-carbon molecule is going to be integrated into the intermediates of the citric acid cycle. Yes. Okay? So, everybody okay with this slide before I jump into the cycle? One final thing to note. How many NADHs and how many molecules of carbon dioxide could I produce through this process here with a single molecule of glucose?
1 or 2,
Why are people saying 2?
because you split the glucose in half.
2 pyruvates. This is only showing you this process for one of the 2 pyruvates that we've produced. So for a single molecule of glucose, you need to double the output of this, which means we would make 2 molecules of acetyl-CoA, 2 carbon dioxide molecules, 2 NADHs, and another 2 protons. Okay? So again, during this process here, 2 NADHs, 2 protons, 2 carbon dioxide molecules, and 2 acetyl-CoA molecules. All right. Now let's have some real fun. Okay. The citric acid cycle, just like glycolysis, looks incredibly scary. But here is what you really need to know. This cycle is going to allow us to make more NADHs and another molecule called FADH2. Where do you think we're going to use those NADHs and that FADH2? Oxidative phosphorylation. Are you noticing a pattern? Glycolysis and the citric acid cycle, while they do make ATP, and this will make 1 ATP every time we go around the cycle, the big gain from these 2 reactions, or these 2 series of reactions, are those electron donors that we're going to eventually use in oxidative phosphorylation, and those protons that we form when we make those electron donors. So, for every 1 molecule of pyruvate, we're going to make 1 molecule of ATP, 3 molecules of NADH, and 1 molecule of FADH2. Here's the easy look at the citric acid cycle. Again, we're going to remove some of the carbons from the intermediates here to produce carbon dioxide. We're going to produce 3 NADHs and 3 protons, 1 ATP, and then an FADH2 from an electron acceptor called FAD. Notice also that it's a cycle because the final product here that we create is going to be used to attach it to those 2 carbons from the acetyl-CoA to fuel the cycle once again. Questions, concerns at this point? Yes, Kate?
You said it will be used by the 2 carbons.
The 2 carbons here will be added on to whatever product we produce over here so that we can perform form the cycle again. And I'll show you that here in a little bit. There's a molecule called oxaloacetate. You won't have to remember. It is going to fuse or bind to those 2 carbons in acetyl-CoA. And then the next 7 steps are slowly going to decompose the citrate that's formed into oxaloacetate. So really what we're doing, guys, is we're building a bigger molecule, and then we're using reduction reactions to remove electrons from that molecule to make stuff that we're going to need for oxidative phosphorylation. And while we're doing it, hey, guess what? We can make some energy too. So whoop-de-doo. Okay? Here is the citric acid cycle. Okay, now, who wants to tell me what I probably won't ask you to remember? Everything. Okay. Any of these intermediate names, I'm not going to care if you remember or not. You can go be graduate students and make it your life's work to memorize those. For my class, that's not what I'm concerned with. This process is used to produce large amounts or larger amounts of NADH and FADH2 than we're capable of producing during glycolysis. And those are going to be used to fuel oxidative phosphorylation. It's also important that this is a cycle, because if it wasn't, then what it would mean, you'll notice, okay, first off, oxaloacetate has 4 carbons, which means when we add the 2 carbons from acetyl-CoA, we now have a 6-carbon compound. Technically, actually, yeah, 1, 2, 3, 4, 5, and 6. A 6-carbon compound. If we were not to have this as a cycle, we would have to use another 2 carbon dioxides to make acetyl-CoA to give us the 6 carbons we need to fuel this every time. The reason it's a cycle is because it's energetically favorable to only have to put in an acetyl-CoA every time you want the cycle to go around than to have to make each and every one of these starting molecules from scratch. Okay? Things that you need to know off of this. First off, if I give it to you in a picture, please be able to identify it. It's the citric acid cycle. Is there an energy investment phase anywhere in this cycle? No. We have an energy payoff here, but there's no technical energy investment, at least none that we've seen. This is a particular type of it, but we're not going to go into it. Okay? The other thing to note, we make 3 NADH molecules and 1 FADH2 every revolution of the cycle. I cannot stress the importance of these molecules enough. Yes, we make 3 NADHs and 1 FADH2 every revolution of the cycle. We make 3 NADHs and 1 FADH2 every revolution of the cycle. And we do make 1 ATP, but I was focusing specifically on the electron acceptors that will then become the electron donors. Yeah. We do also make 1 ATP, and we make 2 carbon dioxides with every revolution of the cycle. So, the citric acid cycle So that really is some of the bigger stuff that I'm wanting you to remember. Remember also, please, with every one of the NADHs that we make, we also make protons.